Hello? Oh, it's off. Oh, let's see. Okay. You're working in the background, so your mic is off. Okay, thank you. Welcome to the meeting. I don't know who else will show up today, but I'm going to go through some things and then we'll, proceed as usual. And if people come in and want to present something, we can do that. Otherwise, we have a good number of things to talk about today. So let's get started. I'm going to share my screen. All right. So first thing is that we have a couple of things. We've got a bunch of materials on the Digital Bacillaria project we're going to revisit. But first we're going to talk about this repository that I found on GitHub, and it's something that I think people in this group would enjoy. Hi, Susan, how are you? You're muted. Oh, okay. So people in this group might enjoy this awesome deep GNN. So GNN stands for Graph Neural Networks. So this is a repository of papers on the topic. I think we've talked about GNNs previously. We've talked about how they work a little bit. We've talked about maybe how to implement them in our group for different problems. This is a nice set of reviews or papers on contemporary topics. So it looks like they're keeping it up 2021, 2020, and before 2020. That's what counts. And Graph Neural Networks is like kind of a historical review. But it looks like it's a fairly new area, so we have a lot of different papers on it. I don't think they have any software. So, I can hear you now.
Yeah.
Good. Okay. So what's new? What's going on?
Well, my one computer decided to quit.
Okay.
My eyes are working better now.
Good.
I have some really weird glasses. You can see that this one has no lens.
Yeah.
So, currently trying to find some better glasses so that I can see better. But, yeah, I've been able to use reading glasses, so now I can do my signal processing. So.
Good. Well, that's good. And everything else is going all right for you? Like your research, you were doing some work on you were doing like a presentation for your seminar or something?
Yeah, I have to do my research project. No, I've had to go back to review my signal processing so that I can do the math for it. Like I know the physical process, and I'll know the equations when I see them, but I know the physical process and I know the equations. I just have to prove the end equations. So yeah, I've got some work ahead of me.
Good. Well, good luck with that.
Fourier transforms, hurrah.
Yeah.
Yeah. That's what I'm doing. So,
yeah.
I don't know. Eventually I'll get there and then I can do other things if I've got that sorted. In the meantime, no.
Yeah.
Yeah, that and our harvest is mostly over.
Oh,
so
okay.
So there's been harvest and a few other problems around here. So, I haven't made as much progress as I should have. Just so you know.
Yeah, it's tough. Like graduate school is pretty tough. Like just trying to keep up with some of the things, trying to master some area and new areas that you haven't really been familiar with in the past. So
yeah, it's been at least 10 years since I've seen this. So
yeah.
Yeah.
Well, good luck. Yeah. So let's see. Go back to this.
I'll turn off this.
We'll go back to this. I don't think there's any sort of like soft. There are a lot of papers that kind of give you a good idea of this area. And so, maybe we should talk more about graph neural networks. I think Krishna is interested. I don't know if he's going to join us today, but that's something we should follow up on. Okay, so then I guess the other thing I wanted to talk about was this Digital Bacillaria repository. So I met with Ujwal and Asmit and Tharun on, I think, Friday. We talked a little bit about this, what to drive this project forward, sort of the next steps. And this repository is something that's a couple years old. We have a paper we actually had a paper that was like machine learning and biomechanics, and it involved looking at Bacillaria as a digital organism. And so this has just recently been published as a book chapter. And so this is the book chapter here. It's chapter 10 in this book, Towards a Digital Diatom, Image Processing and Deep Learning Analysis of Bacillary Paradoxa. So in this paper, to recap, we took Bacillary Paradoxa, which is actually a species that is not very easy to define from the other Bacillaria species. So collectively we call it Bacillaria or Bacillaria spa, which is SP period, which means that there's like this, it's hard to distinguish between the different species in a genus. So in this paper, we mined a lot of data from videos that were available on the web. And we took these videos and we took them apart and analyzed them. So that was Ujwal and Asmit, I think, were mainly involved in that. And they kind of characterized all the cells. So, Bacillaria are these cell colonies that have these long rod-shaped cells, and they move together in a certain way that I think we've talked about in this group. They're part of a larger set of organisms called Diatoms. So diatoms come in many different shapes and sizes. Actually, they're mostly microorganisms, but they come in different shapes and sort of relatively large or small sizes. And so they were able to model these cells in different sort of modes of movement or modes of sort of their geometry. So these rod-shaped cells are stuck together in colonies, and the colonies configure themselves in certain ways. And so when you get a still image, you get all these different cells that are in different positions relative to one another. You have to define the cell centroid, etc. And then, so this is an example of a Bacillaria colony. You have these chains of cells, and they're either elongated or they're stretched out a little bit like this, and you get, they're in different modes of movement. So they're, in this picture, this was O.F. Muller, who back in the 18th century, was able to identify at least 8 modes of movement or 8 modes of sort of this phenotype. And so here's an example here of a colony where there's one cell that's stretched out against another cell, and then the rest of the cells in the colony are aligned with one or the other cell here. So this is an example of stretching, but only local stretching, not global stretching. And then this is a close-up of the rod-shaped cells. They have a morphology. There are a lot of actin filaments inside, and the cell walls are silicate. So they're actually an interesting type of cell that you don't see in, say, like animal cells. So this is the rod-shaped cell here. This is 10 microns for sense of scale, so it's a very small organism. And these colonies are also likewise very small. And this is done by scanning electron microscope. So this is actually pretty high resolution for this spatial scale. And you can see that we have these horizontal slits, the raphe here, and then you have these valves that take in water, and they do other types of liquid and gas exchange, and then you have different structures within the cell. And so they're actually, this is part of their movement. They have these actin filaments that move, and the cells move against one another. So it's almost like muscle in some ways. And so we were able to identify these cells digitally, extract them from video. And then Thomas Harbeck, who we work with, he was able to take colonies and do this analysis on different colonies. This was not a high throughput analysis like Ujjwal and Azmit's analysis, but this is something where he took the cells under a light microscope this time, put landmarks on the cells relative to their extent of their movement against one another. So you can see these cells are extending themselves in the colony, but the cells, they don't come apart. You would think like if they stretched against one another that they'd fly apart and have to reassociate, but they don't. They come to sort of this region where they're sort of in close proximity, but they don't quite come apart. So, they can stretch in each direction like this. And there's this little overlap area here. And that overlap area is, as it turns out, there's a size where the cell isn't overlapping. And so you can define all those parts of the cell that, define the movement. And so you measure the length, you can find centroids, you can do other things. And then you can build a model of movement, which is in a graph further down. This is actually an example of what Azmit and Ujjwal did for each of these video frames for a colony. This is a Bacillaria cell. They found 5 different landmarks on it, and then they plugged it into a deep learning algorithm to find, to do some classification and find bounding boxes. So now you can generalize this method to find cells in any set of images. And so this is some diagrams. This is explaining deep learning, because this book chapter is not for people who do deep learning. So there was a significant amount of this chapter that was devoted to telling, kind of explaining the methodology behind the image processing. So there were some things that were tried and failed. This is like a good example of some methods that failed. So they tried a couple of methods. They tried candy edge detection and watershed segmentation on these colonies, and they didn't work at all. But the method that they finally settled on, which was a deep learning model, Deep Lab version 3, actually worked quite well for this. So this is a good example of how these models and methods kind of work or don't work. And then I think we have some more information. This is actually plotting out some of the features in space. And then there's this graph here of the stuff that Thomas did with the biomechanics, where it's actually looking at the movement over time. So you have your cells, they form this oscillatory movement back and forth. So he's looking at that. They tend to settle down over time in this example. And they form this attractor basin here where you have, if you look at like 2 cells that are moving against one another, you can plot it out as an attractor. And you can see that, like, in this case, this is a nice way to show, and I'll zoom in. This is a nice way to show some of the noise that happens as the cells stretch out and come back together. So as they kind of stretch to their maximal point, there's this sort of noise that happens where they kind of stop and come back. So it's gliding, it's fairly smooth gliding in these parts of the attractor, and then it's a little bit more, there's a little bit more variation on these ends, and these ends are where the cells stop and then come back together. So this is velocity, and so this is like I think the first order of the second order of position, or actually the first order of position. And then you can have, so this is meters per second. And so that's the first derivative of position. And you can get acceleration, which is the second derivative of position, which is going to show more of this sort of noise. And then you can get even higher derivatives of position, which really kind of break out some of this stuff to some of the higher order movement dynamics. So for example, gliding isn't necessarily smooth. It looks smooth here, relatively smooth, but you can see there's some variation in the trace here that we'd like to pull apart and take a look at it. And so, you can do this, but you need higher resolution data than what we have here. The sampling rate here was, I don't remember what the sampling rate was, but it's really, the sampling rate is, your video rate. I think it's like twice the video rate, so whatever a video rate was, which wasn't very high. And, beyond that, you get effects like aliasing and other effects. We actually talked about this, and we did talk about a paper once where you can actually upsample video data, where if you have like a movie that's sampled at 1,000 frames per second, you can sample it at 10,000 frames per second, just so that you can get that extra resolution. So you can do things like this, where you can look at, higher derivatives of position and of motion. So this is the first derivative of position velocity versus position itself. We could redo this plot as like, velocity versus acceleration, which is the second order, second derivative of position, and so forth. And so we can do a lot of these types of things. And then we have, we can also look at the changes between the position of cell 2 relative to cell 1. So if you have a pair of cells that are moving against one another, you can see the relative position as it changes. And so in this case, I think we had a discrepancy here later in the time series as the cell was moving, or as the cells were slowing down in terms of their movement against one another. So this this accordion pattern that you have of movement of the colony, it slows down and even stops. And as it stops, there's some instabilities in the two cells moving against one another. So we can quantify all of this using these methods we've introduced in this paper. So this is in this book. What is the name of the paper or the book? It's Diatom Gliding Motility. So this is the name of the book here, and we're chapter 10 in that book. So that's something that if I can make this paper available to people in the group, it's also available through your library. And so in any case, I wanted to get into the DevoWorm repository, or the Digital Bacillaria repository. This is in the DevoWorm GitHub organization. And so this was the repository that we made for this project when we were writing this up. So there, some of these repositories are fairly old. They're over a year old. And some of these things were, we've had this out for probably as a preprint for probably about almost 2 years now. So, some of the analyses are maybe kind of dated, but, we did, I think for our audience, that was a pretty good, that was a pretty advanced analysis. But we're talking now about going back and actually taking data that's a little bit more controlled, because we took a lot of the data off of the web. So there are different source materials, which made it a little bit hard to do a lot of the segmentation. When you have different backgrounds, for example, in different movies and different experimental setups, it's really hard to build a data set and pull all those images together into a single data set. So that's something that we had to overcome in the first study. In the second study, the stuff that we're going to do now, we're looking at doing some, getting some data from a standardized source. And Thomas Harbeck has been collecting data on diatoms. He's been culturing them and taking videos of them. And so those videos, then we're going to use those, and we're going to, analyze them in similar ways to what we did here. Now, the reason I also bring this repository up is because Hacktoberfest is coming up. So Hacktoberfest is coming up in October. That's why they call it Hacktoberfest. It's supposed to be like Oktoberfest, but they've given it the name Hacktoberfest. And we're going to participate as an organization. And so it means from October 1st to October 31st, we're going to have activities for Hacktoberfest. And really the way this program works, this is through GitHub. This is a program that they've run with different organizations. I think DigitalOcean was the first organization to host, but other organizations host. And the idea is that you go to an open source repository with a badge, and we'll put a badge on the repositories we want to mark. And then if you like do a certain number of pull requests in that repository, you get some sort of recognition or a T-shirt or something. So we haven't given any. We've done a lot of recognition of this, but we haven't given any swag out. So I don't know if we have any swag to give. So I don't know. We'll deal with that. But I think the idea here is to get people involved in it, and maybe give recognition for it and things like that. It's supposed to motivate people to participate in open source. So hopefully we're also doing the DevoLearn repository as well. So if people are contributing to DevoLearn. I think I talked about that last week, where we were talking about some of the people who have been contributing to DevoLearn, and were in the process of producing a new version on that. So maybe if people want to participate in October for DevoLearn, then you'll be part of this new version as well. But this is something that we're going to promote in the next couple weeks. I'm going to put a badge on the different repositories, and I think our repositories then are going to be Digital Bacillaria and then the DevoLearn project, which I don't have up here. But anyways, this is going to be a little bit different type of repository, because it's really centered on some of the work that was done in that paper, and we're trying to do some more work here. Like I said, we have video. There's image processing. There's and I was talking to Ojwal about this. I have some data for people if they're interested in doing this. We may have to set it up where if people want to analyze data, we'll have to create a pipeline for them to access it. So this is going to be maybe that the people who participate in Hacktoberfest are, sort of offline, and we might, give them certain tasks surrounding, like maybe some of the, digital processing, or maybe just maintaining things. I don't really know how we want to work this, but it's a little bit different because most, repositories are, fix this issue of code or whatever. And so they're very clear issues. In this repository, the issues are a little bit scattered, mainly because they involve what we were doing in that analysis. So some of these things, though, I think I'm going to go through these and sort of order them in terms of what we might use for Hacktoberfest. You can label issues here, so I might label it with Hacktoberfest and, give people some guidance on where to go. So I'll be working on that from now until the end of the month, and hopefully on October 1st we can, start off with some people contributing. Hello, Jesse. How are you? I see you're here. Hacktober. Susan said, "What was the title?" This was the title of this paper that I'm showing, or? Okay, so the paper title itself.
Yeah, you already showed it to me.
Oh, okay, yeah, this one.
It's all right. It was toward the digital diatom.
Yes.
And then, yeah, I've got it.
Okay, good.
Thank you.
Yeah. So good. And so the idea of a digital diatom, it's kind of a lot like OpenWorm in a lot of ways. Like in OpenWorm, they're trying to build this digital C. elegans. So the idea is to take the data that exists, and a lot of what we've done in OpenWorm has been like, really based on microscopy data or other types of data like, lineage trees and things like that, and just putting them into numbers, creating models of, say, like neurons, and simulating that. In this case, we're creating, models of cells. So we're doing some cell modeling, we're doing some movement modeling, and we're doing some Actually, as I'll show you in a minute, we're also doing some modeling of sort of I wouldn't want to say the mind, but like something called non-neuronal cognition or aneural cognition. And so that's something that, we are kind of putting the pieces together on that. So we're very early on. And in Bacillaria or diatoms in general are much simpler than even C. elegans. So I think we're moving along on that. We're going to take another pass at this movement and cell modeling, and then we're also going to deal with some deeper issues. And then, hopefully at some point we'll put this all together into something that looks like openworm. And you might ask yourself, what do diatoms have to do with development? Well, diatoms have a very interesting sort of life history and set of growth patterns. They accrete their cells instead of the cells growing like a typical animal cell. So they accrete layers of their cell, and they have a lot of interesting growth dynamics. And then of course they can associate themselves in colonies, which is also interesting. It's very different from what some animals do. Other animals do, of course, form colonies, but they do it in a slightly different manner. And so this is kind of an interesting digital modeling exercise. It always does rely on more data, and it's one of the things about openworm is that we sort of have this idealized organism. You have this adult, or it started out with an adult, and then you have the hermaphrodite and the male. So we don't really differentiate between those in openworm too much. We don't spend a lot of time on the larval stages. It's going to be this digital modeling exercise of modeling an organism. It's like what is your conception of the organism you're trying to model? Is it like just an adult or a static part of their life history? Is it like one stage of their life span? Because some organisms have very different phenotypes, say, in different parts of their life. So those are all things, if you're talking about digital organisms, you want to think about.
The diatoms are an important part of the ecosystem. They're like a base food for a lot of things, including small fish. Like they're something like 20% of the ecosystem relies on them. That's another aspect of them.
Yeah. And they also use them for biofuels a lot. There's a lot of research being done. So they're algae. So they're they live in water. They have this, and so that they're used. People think they can use them for biofuels and other applications like that. Some material science, I think, as well. So they're very, very interesting to a lot of people. There's a community of people who are interested, but like with openworm, it might take on a life of its own, where you have people are just interested in this sort of digital modeling because it looks cool, or it gives them access to a model organism. So okay, so did anyone want to bring anything up while we're sort of at a stopping point here? Did anyone have anything to report?
I just feel like I've been gone for a long time.
Yeah.
And that was due to circumstances, a bunch of them. So.
Oh, welcome back. Yeah. So I wanted to go over this paper then. And I told you about the Bacillaria aneural or non-neuronal cognition. And this title is, it's kind of a working title. The actual title of this paper, and it's almost finished, this is why I bring it up, is The Psychophysical World of the Motile Diatom Bacillaria Paradoxa. So this is, psychophysical world is an intriguing term, because we wouldn't think from what I've told you about diatoms, and all diatoms, but especially Bacillaria, that wouldn't have anything to do with anything psycho. So why am I bringing this up? What are we doing here? So it turns out that this organism, you have organisms that have brains, of course, and then you have organisms that don't have brains. But those organisms behave and they do things that are, there's an analogy to be made with cognition. And so people have made this analogy between, say, like the biophysics of something and some form of cognition. This ranges from like people looking at cells in the human body and building a model of decision making to, people doing things here in this part of the introduction where they're looking at different organisms. So they're looking at all different types of small organisms like slime molds and other types of, organisms that don't have a brain. And so, they're maybe doing things like collective behavior, or they're doing things like responding to stimuli. And so, even though they don't have a nervous system, they're exhibiting something called cognition. And maybe it's worth thinking about cognition a lot more broadly than, say, like the human brain, and think about what cognition kind of means, which is information processing. And so that's not the sort of the Latin root of cognition, but it's what people have come to mean by when they say cognition, is that's information processing. So in this case, you could use a brain, or you could use some sort of cellular structure like this, where the cells have, different components. So it turns out that each of these Bacillaria cells can sense their environment. They can sense water flow, they can sense light, they can sense temperature, they can sense water chemistry. And they can do all of this, and they respond to it. So there's a characteristic response that they make. And so in this paper, we review some of the evidence for this, especially like light sensitivity. And, they can do all this, and they respond in kind. Now, they can do this in a colony, or they can do this individually. In this case, we have a colony of cells that are responding to these cues, and they're doing it in a spatially dependent manner. So, if you're on one end of the colony versus the other. And so, that's part of their decision making, but also that they're connected to one another through this colonial structure. And so these colonies are held together by secreted polymers. So there's this extracellular component that that sort of enables a lot of the dynamics of this colony. So, our Diatom colony is an information processing machine, basically. It's taking in information about its environment. It's operating in kind. It's either slowing down or speeding up its behavior. And then you have all these cells that are acting collectively. So this is an interesting thing, and most people have explained this using biophysical models. So they've used metaphors of explosion, like jet engines and things like that. Very mechanical metaphors, very things that are chemical or biophysical in nature. What we're doing here is we're turning that on its head, and we're saying, well, this is actually information processing. Like something you might see in the brain, but you also see in other organisms that don't have a brain. And there's this whole area of aneural cognition or non-neuronal cognition that allows us to interpret this. So we get into some of the reviewing some of what non-neuronal cognition is. It's basically breaking the behavior down into some of the components of the movements, how those things are put together, and then we introduce models for all of this. So we use this sort of paradigm of psychophysics, which has been applied to a wide range of animals, a wide range of organisms, and it's basically this idea that you have sensory inputs and you have characteristic outputs, and those characteristic outputs can be characterized by some quantitative law or some quantitative rule. So we use the example in this paper of the Weber-Fechner law, which is a law that determines, if you have a certain level of, if you have a sensitivity to changes in a stimulus. So it could be changes in a light stimulus or changes in a temperature stimulus. And the idea is that you can discriminate between two different levels of intensity. And so if you're better at this, you can discriminate between smaller and smaller magnitude of differences. So, I think I've shown this in the group meetings, but, you might have an array of like 10 dots versus 11 dots, or 10 dots versus 100 dots. And the idea is you have to be able to distinguish whether those are the same or different. And you might think, well, that's easy. You just take the 10 versus 100 dots is really easy. The 10 versus 11 dots is really hard, and you would be right. That's true, and that's usually what's found. I don't know, maybe humans use grouping strategies to sort of make this a little bit easier for smaller magnitudes. But in general, they do find this sort of relationship. And so that's kind of what we're drawing from here. But we're also going beyond that and introducing a whole series of models that allow us to look at some of this. So we're looking at Hebbian intelligence and predictive processing. We're looking at some other types of connectionist models. We're looking at signal detection theory. We're looking at something called a collective pattern generator, which is based on a central pattern generator, which is often found in nervous systems where you have a network of neurons and they produce a rhythmic output. So like your heart is a collective pattern generator. You find them a lot in insects and even C. elegans, where you have periodic movements that are generated by groups of neurons. In this case, the collective pattern generator is something that's generated by the colony and different points on this in this extracellular matrix interacting with actin filaments that are moving. And so the movement of the colony is governed by some external stimulus. Each cell is taking an accounting of that stimulus and differences between intensity of the stimulus, and then producing a pattern with this connectionist model that generates this collective pattern generator. And then, also even algorithmic information theory for other types of, looking at different modes of movement and changes in the mode of movement. And then it kind of gets a little bit into dynamical systems to tie that together. I have to finish that part. Well, there are a couple of things that need to be finished. We're pretty close to the end here. And so this is a very sort of broad overview paper. And, it's not like based on it's based on some data, but we don't do any rigorous data analysis in it. So the thing I want to do from this is I want to get some feedback on it from the place where we're going to be submitting it, because it's dreadfully late in terms of where I was going to submit it. And then, we'll keep working on this as a theme in this Digital Bacillaria project. And so hopefully that we'll get some feedback. Is this paper still open to contributions? Yes, it is. Let me send you a link, and I can put this in the Slack as well. And the permission should be open on this. If you want to go ahead and take a look, this is the document. It's a Google Doc, so you can just kind of, if you have comments, please leave a comment. Don't just put in something in the text. It's the way we kind of like manage comments. But yeah, I would appreciate comments, or if you want to add some things in, because we're getting close, but I can't seem to get to the word limit that I need on that, because we've been working on it for months, and I've been trying to massage it for about at least 2 months, and getting a lot of that all in order is very difficult. But anyways, I want to eventually incorporate some of this into the Digital Bacillaria repository as well. So some of these models that we talk about in the paper, these might be something that could be implementable as digital models. So for example, some of the stuff on predictive processing and Hebbian learning, those are things that, we have to kind of come up with a way to model that, some software, some code, or, some sort of simple simulation where you can simulate this sort of thing without having a nervous system. if we're using this Bacillaria model, what kinds of things could we model with this? Or how would we build a computational model that would demonstrate this without a nervous system? So what's interesting about predictive processing is that it's based on this idea of free energy minimization, and there's a bunch of literature on this. I may need to rewrite this part a little bit, but it's this idea of active inference, and there's this idea of free energy minimization for cognition. And this actually ties in well with the free energy minimization models that we use in biophysical movement. So if we just used a biophysical example, we would have a biophysical model where there's energy minimization that tells you what the behavior should look like, and then you would have this more sort of cognitive or information processing model, which would do the same thing. So I think it's interesting how there are parallels there that you can build upon. And then the free energy principle, if you use that, or you use predictive processing, which is related to that, those sorts of things then give you sort of a pathway into other sort of more cognitive models, where you can maybe say that there's information processing going on. So you can use these metaphors in some of the computational mechanisms to draw parallels and bridge between things that don't look at first glance to be related. But anyways, ultimately, we'd like to put this into the Digital Bacillaria repository as well. So we have a behavior section, but there isn't much here. It's just some images and files that some code that was put in here, but there isn't much structure to this yet. So we're really trying to work on this part to fill it in, and then eventually we're going to have this as like a resource for people. So this description is dated, but I think we've got a lot of stuff. I need to update this. This was, I think, created at the beginning of the project. So yeah, we're going to be, Ujwal wants to revive this, Asmit wants to revive this. I also talked to, who else did I talk? I think Akshay wants to do some work with this as well, but I'm not sure. We're going to kind of go over this in upcoming meetings and kind of hash out some of these things. And then we'll be doing things for Hacktoberfest in this as well. So that was a lot of talking about that. But I hope people we can pick up activity on this again. Maybe next Google Summer of Code, we can actually get this funded. So last year we had a project on this. We had a lot of applicants, but we didn't get it funded. So we had 7 or 8 applicants for it that didn't get anywhere with that. So, but they did actually come up with some really interesting proposals. So we'll follow up on this. I wanted to go to the submissions page, the document that we have. And again, we have all these things that are kind of hanging in the air. We have this boring billion book that, this early life, embryos in early life idea that we've kind of yet to realize anything with. We have this Kindle book idea, which is also kind of hanging out there. We have a mathematics of DevoWorm. That's something that I talked about. I think last week I talked about sort of the mathematics of OpenWorm and showed that document where we had the equations and we had some, methods. That's kind of what we're looking for this, but for DevoWorm. And so if you want to find out more about that, go back to the video from last week. I did put it in the Slack channel, so you can check that out. There are also some opportunities to do some molecular level simulations. So I was talking about diatom motion and the derivatives of position, derivatives of motion. That's something that so jerkiness is where you get really high derivatives of motion, and it shows that sort of variation at a very high scale or high level of, so you can see a lot of movement variation if you look at something called jerkiness, which is like, position or like, distance per second per second per second. And that's, it's interesting because, a lot of this higher level sort of variation, especially when these cells change their mode of behavior, their position is really interesting. So we have a couple of lines on that. I don't know where that's going though. And then finally, we have this Neuromatch 4.0, which the submissions are coming up. Actually, it's October 25th instead of the 15th. But this is this conference, this open neuroscience conference, computational neuroscience conference, that I would like to submit some abstracts to again this year. Last year we did some stuff on diatoms. We also did some stuff on artificial neural networks and biological neural networks. This year, actually, I was thinking maybe we could do something more oriented towards DevoWorm and kind of maybe something where we have an event during Neuromatch, which is in December. The actual conference is in December. And I don't know if people are interested in this, but this is something that we could do where we have like a meetup of people, maybe even across to OpenWorm. I might bring it up at the annual meeting later this month. But to have like a meetup of OpenWorm people and to have like, it could be informal and virtually, or it could be where we present on our different projects. But it would be to this Neuromatch audience, and it would be kind of tied into Neuromatch. So, I definitely like to submit some abstracts, and if you have anything you'd like to submit, let's talk about how to do that, how to make that, interesting to this audience, and, maybe we can make some DevoWorm tie-ins here. So yeah, I think they're interested in a lot, like there's a track on cellular and molecular work. There's also a track on some other types of computational work. So there are a number of tracks we could fit into, computational tracks for machine learning and deep learning. So there are a number of opportunities here. And then, of course, the NeurIPS workshops, and NeurIPS is coming up in December. So if you're interested in machine learning, there are a lot of opportunities to learn from that. I think a lot of the workshops, their submissions are closed, but we should check this out as well.
I have a few ideas about Neuromatch.
Okay.
I think it would be good to do what you said about the OpenWorm, like everybody to present what they're doing. And I think as far as specific presentation, either by, I think that would be good in general to do like a bigger thing, but also I would particularly be interested in doing something along the lines of I don't quite know how to phrase this yet, but it's something like a tour or display of either model organisms or just the merits of using simulations to do some stuff. Like a lot of our work in here and in the other group is simulation based, or using these sort of things like I think there could be something, because I'm thinking a little bit back to I didn't quite this is something that never happened at Princeton Envision with Logan Thrasher Collins, who was very into like, at the time he's sort of shifted on from that now, but at the time he was very like in favor of getting to like being able to fully model like the nervous system and the biology of a bee as opposed to a fly, and like how being able to make advances in modeling and simulating those organisms could help us. And also like there's various things we've done in terms of that are already like why look at non-neuronal cognition, why look at these things that we can model in different ways, and some kind of a thing along those lines. I don't have it fully fleshed out, but like sort of a maybe not necessarily like I could see this is very much a typical like Jesse thing to want or say. A lot of my projects fall in this category, but like, you know, what's sort of the history of this? Where are we at right now, and what will be good in the future? Like, what's the cutting edge of this, and what can I provide? This kind of a thing. This is a very like, all of my presentations have that theme anyway, but like just in the context of the simulation model organisms, that comes to my mind. And I don't know if I imagine there's been things that have already been done here at OpenWorm or DevoWorm, but like, that's just what like, that's what comes to my mind as, yeah. That's what comes to my mind as, it's a very Jesse thing to say and suggest, but you know, I'm curious what fits with that.
Yeah, so I think actually the model organisms part is interesting because I know we have the DevoZoo stuff, and that's of course something we can always go back to and you leverage. But I was at a workshop this summer where it was like biologists and modelers, and one of the things that people were, some of the modelers were really intrigued by is, all these model organisms that the biologists talk about. Like, they have different model organisms for different systems, and I think last week I had a paper, I think this was in the other group, where where they looked at organoids and other model organisms, and they looked at how they like sort of recapitulate biology, which means, I guess, be close to something like human behavior or biology. And so the biology, they made this assessment of it, but of course it was like based on some subjective criterion where, you would say, does it, recapitulate human biology in what way? Disease or development, or, maybe has a similar set of genes controlling some process. So those are all things, and I think that idea of model organisms, I think, is something that there are very few resources on, like, kind of reviewing model organisms and their usefulness. But especially like when we get into the digital organism space, where, like, if I want to build a digital model of this organism, first of all, how is it useful? How does it fit into, like, other model organisms? If we had a zoo of these, could we understand a little bit more about, like, the diversity of biology, or, different processes? Like, if we had different models of, like C. elegans and bees and, fruit flies, could we understand some process in common? We actually did a paper on that several years ago, where we looked at, like, developmental data or tracking data of embryos, and looking at, like, some of the potentially common processes across different types of embryogenesis. So, I mean, those sorts of things, you can make comparative things going, you have comparative work going on there. And so it's very interesting. I think we should try for that. We'd have to kind of define the scope of it a little bit, and then, find sort of the most interesting take, because it's like a 15-minute presentation. So what are the takeaways we want from that? But I think it's definitely good idea.
Yeah, I would be curious, like, if you want to, maybe you could, like, refine what I've said, what I brought up, and what you said, and, like, mention it in the Slack, and, like, tag me in it, please, because I'm fleshing out, like, this is an on-the-fly thing, and I was like, oh, that would be cool, and I'm definitely interested in that, because I feel like I haven't done anything really with DevoWorm for a while, and that would balance out some of the things on, like, that'd be biologically based with a lot of the things I'm doing. So that'd be pretty cool. Even the version that you said. The other thing I would like to mention, if we have a few moments, I put in, and it could actually be related to a Neuromatch related thing too that we could do, but it's kind of it's in the camp also in orthogonal, just to kind of, like, show this the Brains Through Time Reading Club. I'm not saying we can initially do this in terms of a Neuromatch related thing, but just to kind of show it. It's about the evolutionary history of the vertebrate brain and discussion between just neuroscience and so on, and all these, these people here. And it's starting in October, so it wouldn't really be like a lot of time. I think you're going to do weekly meetings, so it would be sort of like, it would be later on and a shorter time thing to do, but I could see I'm maybe interested in presenting something like discussing this at Neuromatch in some form, or like making it a direct, like making an adjacent presentation that would go along with this, and kind of thinking about the Paul Kaisik stuff that we talked about in Johan's group. Mostly, I think not. I don't think I mentioned that in DevoWorm, but just sort of that sort of evolutionary stages of development for how brains and nervous systems have come across. So again, I don't have a specific project set in mind, but the two things that immediately come to mind about Neuromatch and DevoWorm are what we just talked about, and then something like this. And whether or not I'm probably going to do this group like 100% either way, in combination of some things going on with orthogonal and just cognition features in general, but also like I think there's a lot of fodder for making an interesting presentation at an event like Neuromatch or something like this too. So that's on my mind about it.
Yeah, I think that's also, that's something that would be also another Neuromatch candidate. Something on this. And then, something you might learn from this, like, we could think of a good topic that we could explore in a 15-minute talk. There's so much there. And, it's, you don't have to wait for the reading club. I guess you could just go and look at some of the resources that they have, and, maybe this is an interesting idea or that's an interesting idea. And then, you write up the abstract, and then as we get closer to the presentation, then you have more to flesh out as you've been thinking about it. And so that would work too.
I partly know why axolotl salamanders are a model organism.
Why?
I did a presentation in Japan on it, so.
Oh, okay. Yeah. Yeah, we could do, I think, like, kind of doing some model organisms, but, you have to find the right hook there. It's like, just a tour of model organisms. People kind of are somewhat aware of them. Like, especially neuroscientists, neuroscience students, they're kind of aware of the model organisms, but, like, how much are people aware of why they were selected, or, how they're related to one another? That's the kind of thing maybe that would be useful, because I don't think a lot of people appreciate that. And I think there's this missing link there between, like, I'm using this model organism and, this is why it's important.
Am I still here?
Yeah, you're still on.
All right. The only other thing that comes to mind about Neuromatch, and I was going to mention this in the other group, I'll mention it here because it's maybe more biologically based, is there's a lot of interesting discussion about the Grossberg book that came up with Johan and other stuff like that. So just wanted to mention, whoa, what is going on here? Just wanted to mention that. I'm not saying we do like a book review, but more like just mentioning that for the sake of it. I'll leave it at that.
Yeah, thanks for that. That was a good overview of maybe some things we can do here, because it's kind of what I was looking for. I wanted to see if people were, we could, I know probably Krishna might have some ideas too, but anyways, yeah.
Yeah, I'm like finally reclaiming my mornings, and I know like our Saturday morning meetings are I'm there, but like having functional mornings during the week are like I have to do that now because of all the stuff on my plate, and I'm just like, I'm here, I can check in with DevoWorm, and there's a lot of cool stuff, and so Yeah, Neuromatch is on my mind a little bit, and I think between this group and orthogonal and everything else, I think it'll be a lot of fun, and I think it'd be a really good target to center some of our upcoming projects. And especially for me, like I've been really out of the loop with DevoWorm for pretty much, until my life. For those, I haven't really said it much in this group, but like my life changed a lot in the last 6 months or so. So like being here in DevoWorm again is like, ooh, like, okay, yeah, I'm definitely curious about the model organism / simulation stuff that I mentioned. And in general, like even for like Neuromatch is kind of a nice like target because you can, hey, we can make this and do that and make it it's a very clear objective to do. But in general, I'm interested in stuff like that. So I'll just note that and state my interest and so on.
Yeah. Very good. So finally, to wrap up the meeting today, I'm going to go over a couple papers. Let me go back to the... okay, so we're back here. Okay, so I guess I have a lot of things in this folder, and actually I was going to talk about two things. I'll be very Connectome-centric today. So today I'm going to talk about two papers. And this is first one is linking the connectome to action, emergent dynamics in a robotic model of C. elegans. So this is something that OpenWorm does with our robotics group, and we're doing stuff with simulating the connectome. And this is linking the connectome to action using a robot. And so this is a group, Valencia, Urbina, Canas, and Gleiser. And they're from Argentina. Okay, so they have I don't know how prevalent they are in the literature. I don't think so, but this is something that they did here. We analyze the neural dynamics and its relationship with the emergent behavior of a robotic vehicle that is controlled by a neural network, numerical simulation. So this is not like the kind of thing that OpenWorm is doing with, modeling ion channels and picking specific cells, but they're actually looking at doing a numerical simulation based on C. elegans nervous system. So, this is a very popular connectome to work with for people, because it's got a finite number of cells. We know what the cells do. They have, very well-defined circuits that actually give you good behavioral outputs. So this is a common thing, and people aren't, simulating the connectome of other organisms as much to build behaviors, mainly because it's harder to do. With C. elegans, you have a lot of factors that work in your favor.
Okay.
So, you're going to say something?
Oh, no. I was, nope.
Oh, okay.
Nope. Nope.
The robot interacts with the environment through a sensor that transmits the information to sensory neurons, while motor neuron outputs are connected to wheels. So this is actually a lot like the Bratenberg vehicle, if you're familiar with that. In my other group, we talk about this a lot. It's basically the idea that you have like a vehicle with wheels. The effectors are wheels. The sensors are the sensor. They're usually up at the front. They're kind of like eyes, or maybe something that feels the environment. And so you can make this connection within the robot. And actually, Bratenberg vehicles are very small connectomes. They're not even connectomes, in fact, a lot of the time. But they actually can produce interesting behaviors that are very similar to emotional states. So it's just interesting aside that you can do that with a very small number of connections, and with a sensor and effector system like that. This is enough to allow robot movement in complex environments, avoiding collisions with obstacles. Working with a robotic model makes it possible to keep track simultaneously of the detailed microscopic dynamics of all the neurons, and also register the actions of the robot in the environment in real time. This allowed us to study the interplay between connectome and complex behaviors. We found that some very basic features of the global neural dynamics and their correlation with behavior observed in the worm appear spontaneously in the robot, suggesting they're just an emergent property of the connectome. So, they talk here about small connectomes or small nervous systems, Drosophila, which is the fruit fly, the zebrafish, and C. elegans. And the reason we're interested in small connectomes is because they're tractable in the sense of analyzing them, especially for generating behaviors. So this C. elegans stands out as the first animal whose complete connectome has been mapped. And that's not entirely true. There's some other small connectomes. I guess there are some small connectomes, like parts of other brains, that have been mapped out. And there are a couple other small connectomes that have been mapped out that are even smaller than C. elegans, although the range of behaviors isn't as rich as C. elegans. But we can talk about that later. So they talk about this. And so what's also interesting about C. elegans is that it has 302 nodes or neurons, but you get this highly specialized structure. So you get these network motifs, which are common patterns of connection that lead to different types of outputs. So you might have like a wheel and spoke motif or a triangle motif, where the nodes form through their connections form some pattern. And that usually has some functional significance. They also have things called community structures, which are just how the neurons are connected together. Rich clubs and small world network structure, which have to do with whether there's like a controlling central node that connects to a lot of other nodes, or whether the connectivity is random. And it turns out it's not random at all. It's very highly structured. And so there's the small world network structure, which kind of governs a lot of the aspects of C. elegans connectivity. And small world networks are interesting because they have what they call a short path length across, which means you only need a couple of hops to get across the network. But they're actually they're also highly structured, which might suggest the opposite, that it would be harder to get across the network quickly. But the small world network structure finds a happy medium between the two. So there's a lot of structure in these connectomes, and they kind of go through the literature here on this. Here are the results here, where they have looking at emergent neural dynamics. So they use a firing threshold, and they use again, I don't think they're sharpening the neural activity in terms of different ion channels, but they're actually using a model of like threshold activity. And so they're able to look at the outputs of these cells, different cells, and they're able to look at do some signal processing, as Susan is becoming familiar with, and they're looking at the output signal and the frequency of the output, and then they do some principal component analysis to look at this. So they analyze their network, then they look at some of these nested neuronal dynamics, where they're looking at collective oscillations, and they're looking at how those collective oscillations are coupled in the network. So we talked about with the Bacillaria, you have these networks of nodes that produce oscillations that are stable, and that's what they're looking at here. But in this case, neurons will produce oscillations. They can produce them autonomously, but then when you connect them together, they result in more complex patterns, more complex states. And so that's what they're doing here. So you could have like a single neuron that's oscillating, and then another neuron that's oscillating, and they can become synchronized, or they can become coupled in some way that shows a number of different stable modes. So these are things that if you're familiar with fireflies and the firefly model of synchronization, where fireflies flash their luciferase sac, when they'd use it as a signaling mechanism to signal other fireflies, and over time they can synchronize their flashing. so that all the fireflies are flashing at the same time. And so this is a similar thing that you see in neurons, whereas they're connected together, they synchronize or they order their oscillations. And so this actually can result in some very ordered behaviors. So you can observe these nested neuronal dynamics from the head of the C. elegans to the entire nervous system, including the ventral nerve cord and tail ganglia. And then they show some more examples of nested neuronal dynamics here. I'm looking for some of their robotic work here. I guess they're now extending this model to global dynamics as it relates to action. They're doing some dynamical systems modeling here, where they're looking at this activity in a couple of principal components. So they're able to take this activity and decompose it into different movement patterns. So you have backwards movement, forward movement, rotate left and right movement, and then a stopping movement. And they're able to pull this all out in this principal component. So they're able to take the neural information, put it into a principal component analysis, and then map it to some behavior. So this is largely, this is something that's based off of Tim Busbus's work. So Tim Busbus used to be a part of OpenWorm, but he parted ways with OpenWorm, and for a number of reasons. And they've been using his program, and they're running it on a Raspberry Pi computer. So this is interesting. They're running these very simple to build robots. Then they're able to do this work. So this is very interesting work. The other paper I wanted to talk about is this Projectome paper. And this is a very long paper in eLife journal. And this is on a projectome of the Bumblebee central complex. So this is another one of these smaller connectomes, but this is actually, part of a larger connectome. So this is in the Bumblebee, which I think Jesse talked about with respect to Logan, one of his friends from another group, and is interested in modeling the Bumblebee. And so in this paper, they're actually talking about getting towards sort of characterizing the connectome of the Bumblebee. In this case, they're actually interested in what they call the projectome. So the abstract reads here: insects have evolved diverse and remarkable strategies for navigating in various ecologies all over the world. Regardless of species, insects share the presence of a group of morphologically conserved neuropils, known collectively as the central complex. So this is a part of the brain that is, it's this, I guess neuropils are a certain type of, it's a thing in neural development. So I'm trying to think of an easy way to characterize it, but I can't right now. The CX is a navigational center involved in sensory integration and coordinated motor activity. Despite the fact that our understanding of navigational behavior comes predominantly from ants and bees, so again, our model organisms are limited in insects. So navigation in insects we know mostly from ants and bees, and there are a lot of different insects and probably a lot of different modes of navigation behavior, but this is where we study it most. Most of what we know about the underlying neural circuitry of such behavior comes from work in fruit flies. So, in other words, we know a lot about ant and bee navigation, but the neural aspect of this comes from fruit flies. So we don't even know that much about what goes on in the minds of ants and bees, but we try to make that extension from fruit flies. And of course, that's not a very good model because there's a lot of uniqueness amongst these different groups of organisms. So that's one of the drawbacks of a model organism. Here we aim to close this gap by providing the first comprehensive map of all major columnar neurons and their projection patterns in the CX of a bee. We find the numerous components of the circuit that appear to be highly conserved between the fly and bee, but also highlight several key differences, which likely have important functional ramifications. So they look at these navigation behaviors. They're looking at the different species that exist, and they're looking for different things that are conserved and different things that are unique to each system. And so this kind of gives us this, so it gives us this model of, what to look for. They're looking what they call the Projectome, which is looking at these neural pills, which actually are parts of the brain. And these neural pills are highly interconnected by the arborization of the three classes of neurons. First, columnar cells are central to the most computations carried out by the CX. They link the four adjacent CX neural pills and provide the basis for intrinsic computations, but also generate output to other brain areas. And so this kind of gets into some of this, what this connectome looks like. It's quite a bit different from C. elegans connectome, and that it has different structures. It has different types of neurons. The neurons are all sort of determined by genetics and by the lineage tree as tightly as it is in C. elegans. But so it's a different type of connectome. And then the question arises how much of this Drosophila connectomics data is representative for other species, and how much of it is specific for the fruit fly and its ecology. So, some of what Drosophila has and what we study is actually very specific to their ecology and what they're doing behaviorally. Now, we don't know, like for C. elegans, the same is also true. If you go to other nematodes, you see other vastly different types of nervous systems and connectomes. So what we're learning from C. elegans connectome is nice, and it maps the behavior cleanly, but it doesn't tell us very much about anything outside of the specific ecology of that organism. So to address this issue, a counterpart of the Drosophila CX connectome will be needed. So they provide a first step in understanding this, though, and it looks like the Projectome is something that is a term that's very specific. It's a type of connectome. They introduced this term in Kesturi and Leichman, and I guess it's just a matter of looking at how things project from one part of the brain to another and using that as the basis for a connectome. So your connectome can take on different, you can construct your connectome in different ways. One way is to just take neurons and look at the connections between them, whether they be, through synapses or through something like gap junctions, which is the way that the C. elegans connectome has traditionally been constructed. Or you can take this sort of projectomics approach, where you look at how one area projects to another area. And in the human brain, that's how a lot of track tracing studies have been done, using this idea of one area projects to another, and so that can tell us about function. And so this is a very long paper. It has a lot of good images in it. I'm going to skip that for now. This is a nice set of images here where you can see that, you have these areas, these neural pill, and they're projecting to other areas. And you can see that they're looking at these projections here and how they're laid out. And so they're using different dyes to mark these connections. So, this is the kind of method they use when you have a really complex connectome, is they have to use tracing dyes to look at some of these connections and see where they go. Because it's not immediately apparent from just doing a high resolution microscopy image. So this is how they do connectomics in more complex nervous systems. So that's all I'm going to talk about today in terms of papers. I hope that was useful. Looks like Susan's the only one left. Okay, so Assaf has to go. Thank you for attending. And looks like Jesse had to drop off, but all right.
What was the Raspberry Pi reference there?
So yeah, that was for this paper on the C. elegans robots. So they were building robots using a C. elegans connectome, and they were actually using Raspberry Pi technology to simulate the connectome. Let me see if I can find. Well, I have the papers here.
This what was author title?
Yeah, here's the title. I think it's an archives paper, but I'll put the title in the chat here.
Oh, okay.
So that was the paper linking the connectome to action.
All right, I am taking notes today. I finally managed to get the screen copy going.
Oh.
Anyways.
Yeah.
Okay. All right. Yeah, I've just been sitting listening. It's nice to listen instead of being on the spot.
Yeah,
Yeah. Seems to happen to me a lot.
yeah, it is. It does. It's okay though.
Yeah, anyway, so I'm here and I just thank you.
Yep.
I'll try again next week.
Yeah. Look
Yeah,
forward to
my
it.
operation for my eye was on a Monday, so I missed that. And then I remembered that there was such a thing as DevoWorm, and then it was on a Tuesday, and then on the Tuesday my computer blew up, and there's been a couple other problems.
Yeah.
So yeah, so I'm sorry I missed a month. I think.
That's okay. Yeah, we're always talking about different things, so it's but that's fine whenever you can make it. So yeah.
Okay.
Well, thanks everyone for attending, and if you're on YouTube, talk to you later. Next week we'll be talking about different papers, and if people want to present, you can go ahead and bring whatever you have to the meeting. And
Okay. Well, I still have to work on a couple of things for you, and I did find point cloud. Let's see. Tom Portuguese gave me information on a free program you can get to do point cloud. So I'll maybe send that information to you.
yeah, that would be great.
Okay.
All right. Thank you for
All right.
attending. Have a good week.
Okay.